Hi, everybody, and thank you very much for your interest in this topic. I'm going to take the opportunity to perhaps talk in rather general terms, as this is probably quite a mixed audience of people with lots of different interests in this area. So I sort of changed my talk slightly. I was going to talk about some of the technical aspects of how we model disease, but I think that might be a little bit sort of dry. So I'm going to talk in general terms about why it is that human beings might be subject to a disease called amyotrophic lateral sclerosis, or motor neuron disease, which I specialize in. So I am a clinical neurologist, and I see patients with degenerative disorders of the nervous system, principally amyotrophic lateral sclerosis at John Radcliffe Hospital in Oxford. I also run a laboratory where we try and model the disease using stem cells from patients and other laboratory models. I do research in patients and in clinical trials. And I'm also head of the Nuttall Department of Clinical Neurosciences, which is a very large department in the university devoted to understanding the brain in health and disease and how we might improve human well-being by treating some of these difficult diseases. So let me start with one of the problems of our time. So, we could spend all the time discussing what's more important: global heating, infectious disease, clean water, or the fact that the populations are aging. This is a problem which is not just a first world problem, it's one which really affects humanity in its entirety. And in large populations in Asia and Africa, which are rapidly aging, diseases of the aging nervous system are becoming an increasing problem and will in the future be a dominant problem for society everywhere in the world. So if you simply look here at the trend in the last 150 years for life expectancy, it's only gone in one direction. And that leads some people to think that if we carry on like this, we'll all live for hundreds of years. I think there's a limit on human lifespan of about 120 probably, for complex biological reasons. But at the moment, we've still got probably quite a long way to go. And you can tell that the projections that we have in these countries here are that in about 40 years' time, the median survival life expectancy in the UK would be about 90, and other equivalent countries. The USA, curiously, lags somewhat behind other so-called developed countries for, I'm sure, interesting reasons. But the fact is that people born today are very likely to know their great-grandchildren. I didn't even know my grandparents, actually. 3 out of 4 of them died before I was born of heart disease. And in about 2050, there may be as many as 5 million people over the age of 80, and a significant proportion of those will have dementia and require care. And at the moment, it's calculated by the Alzheimer's Association, the annual cost of caring for people with dementia in the UK is more than 25 billion a year. That is more than was ever spent in the 20th century on researching Alzheimer's disease as a condition. So our annual spend is far greater than what we spend on research in a single year or in a century. And that tells you that the priorities of society need to be reorientated if we're going to try and solve this kind of problem. So at the moment, if you take the 3 commonest neurodegenerative diseases, and I'll define that term in a minute, there are about 7,000 people in the UK living with motor neuron disease, 10 times as many of those with Parkinson's, and 10 times more with Alzheimer's. So there are, in the region of 750,000 people living with Alzheimer's disease in the UK at the moment. That's a huge number. Each one of those requires caring for, either by a team of employed carers or by their families, who are then taken out of economic activity. So these problems are massive for society and essentially unsustainable unless we can find some solutions. So here is, just to bring some human narrative to the problem, the story of a typical patient who comes to my clinic. This man's 55, he's at the prime of life, he's physically very active, he has an active job, he plays sport. He noticed that over a period of months, his left upper limb, so here you can see his left upper limb, it looks wasted compared to the other side. He noticed he was getting weak. There was no pain, And he gradually found it more and more difficult to do his job as a plumber and came to see his GP, who referred him to a neurologist, who referred him to me. And this is a man who, when I examined him, just 6 months into his first symptoms, had already got physical signs in his limbs and elsewhere that I could see that told me he had a diffuse abnormality of his nervous system that was a degenerative problem. He went on to develop weakness in all 4 limbs within a year, meaning that somebody else had to look after his daily needs. He required ventilation at night because his breathing was becoming weak. He required a feeding tube into his stomach because he was unable to swallow, and he died 24 months after his first symptoms. So you can see this is a highly aggressive malignant process. And the key thing here is this person was extremely healthy, perfectly well, in the prime of life, physically active, and then within 2 years, he had died of degeneration. So what do we mean by that? We mean that a nervous system which is developed completely normally, so that somebody's functioning in a completely normal way, undergoes loss of structure, meaning the nerve cells lose their connections, and function, because those nerve cells and their connections provide function. In this case, the function is voluntary movement. Now, this is characteristic of degenerative disease, a slow and inexorably progressive process without any explanation. If you do a scan of this man's brain, you wouldn't see anything at all. It would look normal, because what's happening is happening at the microscopic level, where the connectivity of the many billions of nerve cells in the brain is starting to fail. And if you look down a microscope, what you see is that nerve cells that have died, so this is the post-mortem of a patient with motor neuron disease or ALS, you can see that there is protein, this brown stuff is accumulating in the cells, it's stained with an antibody, and for each of those diseases I mentioned, ALS, Alzheimer's, and Parkinson's, there's a characteristic neuropathology occurring. So there's something very different about each of these diseases in terms of what's happening inside the cell. What unites them all is that as you get older, your risk of these diseases goes up. Okay, so what are we talking about here? We're talking about a system, because this is not a condition in which the whole brain degenerates. And this system is called the cortico-motor neuronal system, meaning it starts up in the motor cortex and it goes down into your spinal cord. So there's a whole bundle of nerve fibers here, about 1 million of them, which receive input from voluntary centers. I decide I'm going to move my fingers, these cells are activated, they send the message as a group of fibers down to the spinal cord in my neck, and these cells in the spinal cord, also called motor neurons, go out into muscle. So for me to do something simple like this requires many hundreds of thousands of individual computational events in nerves. And these are the things that disappear as the disease develops and why you might lose function. It also explains why simply trying to repair this by throwing a few stem cells into the nervous system, by injecting them in the spine, as has been done, is extremely unlikely to reconstitute function, because that function depends on this extremely complex network of interconnections. Now, if you look in, we start to ask the question, as we are this afternoon, why are humans vulnerable to ALS? So this is not a disease that, as far as we can tell, exists naturally in the animal kingdom. Now, that's a difficult question, because if there were whales out there in the ocean that got motor neuron disease, I don't think I would necessarily be able to tell. Similarly, giraffes, I choose these animals because they have very long nerves. Giraffes, are likely to be eaten before anyone gets to them to find out why they died if they start developing weakness. However, there are zoos and there are lots of zoologists looking at animals, and as far as we can tell, nobody's come up with a convincing case of motor neuron disease in another animal, including primates like chimpanzees. Now, this tract I mentioned here, this corticospinal tract, over evolution has acquired many, many more fibers. So let's just take chimpanzee and human, which, in broad terms, we are very similar creatures. We have the same kind of basic anatomy of fingers and limbs, and yet we have 30% more fibers in this tract compared to chimpanzees. And it turns out the fibers that we have acquired in the last 6 million years since we last had a common ancestor with chimpanzees. These fibers seem to be direct connections. So they are actually not going via other nerve cells. It's direct connection from the cortex down to the spinal cord. And perhaps that direct connectivity is part of that's a system which allows tremendous manual dexterity, but perhaps that comes with some price in evolutionary terms, a vulnerability in the system that might ultimately lead to something like motor neuron disease. And nerve cells are really architecturally extraordinary things. They are extremely long fibers with projections. So this is a rat spinal cord into which somebody has injected dye into a motor neuron. And you can see all of the different projections from that one cell. In fact, the main projection goes out into the rat's limb, and the equivalent cell in our spinal cord goes out into the bottom of our leg and down to our toe. It's roughly a meter. The cell itself is 30 microns, 30 millionths of a meter in diameter. So it's basically 30,000 times longer than it is wide in the cell body. And the reason I say that is because the bit at the end, down at your foot, innervating the muscle, that is the actual bit that causes the function. And that's very far away from the cell body. So these cells are really extraordinary in their architecture. And they have many things about them which make them vulnerable. They have to have specialized transport mechanisms to transport the right protein and things right down the cell. They undergo all sorts of other changes. And principally, they don't divide. So if I give blood this afternoon, it would be replaced within 24 hours from my bone marrow from stem cells. If you lose a nerve cell, it doesn't get replaced. The other thing about neurodegeneration is that it obeys compartments. So what I mean by that is that if you take motor neuron disease, this particular corticospinal tract and its connections is really what is affected. I mean, at the end of the disease, it is correct that at post-mortem you can see some changes elsewhere. But really, this is overwhelmingly a disease of one anatomical and functional compartment, the one devoted to voluntary movement. If you look at Parkinson's disease, it's a different compartment. It's the system devoted to posture and spontaneous movement, actually involuntary movements in general. So unconscious postural and other movements that we make. That's what goes in Parkinson's. It's true that later on, cognition is often affected, but this is a condition which doesn't just explode across the nervous system all at once. These diseases obey compartments. The other concept which I think is really important is that about 10% of the patients I see in clinic with motor neuron disease have a genetic mutation or variant, which is the cause of their condition. But remember, they've had this genetic variant from conception, and they have gone through many cell divisions to form the organism over many years without that mutation causing any kind of problem at all. And then within a short space of time, those patients who have a genetic mutation may go from a state of normal health to one of catastrophic decline and ultimately loss of life. So how does that happen? That tells you that it's not, the genetic mutation is not a sufficient condition for getting the disease. You have to have time as part of the equation. And if you plot the log of the incidence of a disease with the log of a different age, so the age of 30, 40, 50, etc., what you get is a straight line. And to epidemiological statisticians, this means that this is a multi-hit process. You acquire hits as you get older, and that the number of hits required to cause the disease is equivalent to n-1 the slope of this. So this is a technical statistical matter. You have to take my word for it. But basically, it means that there are about 5 or 6 individual biological events that are required to trigger motor neuron disease. And this is rather reminiscent of cancer, where again, you have multiple hits before you get the disease triggered. So then amyotrophic lateral sclerosis, its nature is that you are born with some kind of genetic background, you develop, and perhaps errors occur in the way that you develop. You might acquire other mutations when cells divide, other errors which might seed degeneration, but you have to get older. And that is what we have to work out, the intersection of aging with all of these susceptibility factors. So one way of looking at this is to say, well, who gets ALS? So in the clinic, we have, and we're not alone, colleagues around the world have had the same observation. when you see hundreds and then thousands of patients with what for most neurologists is a relatively rare condition, things start occurring to you. So it occurred to us that most of our patients don't seem to have many other illnesses. In fact, we would often joke with medical students that, this is a very thin set of notes. The patient coming in next, that means there's a high chance that the diagnosis of motor neuron disease is correct, because they typically don't have lots of other conditions. They don't have diabetes or heart disease or cancer. So are they physically fitter than the background population? So once you start tuning into this in clinic, you can easily have what's called confirmation bias to confirm your prior assumptions. You've got to be a bit careful about that. But when this is studied, a story starts to emerge. We see lots of people who have been athletes at a high level, marathon runners, or have been in the military service. We see plenty of people who haven't, but there is a sort of trend. And it tells you that maybe people who get this disease are not a random selection of the population. They have certain characteristics as a group. In an individual case, that may be much more difficult to discern. But as a group, we get the sense that they're not random. Now, there are lots of different genetic variants which might lead to motor neuron disease. Each of these little jelly beans here is a gene for which there's some evidence, often quite strong, that if you mutate it, you will get motor neuron degeneration at some point. So it just tells you it's extremely complex. This is what's called a Manhattan plot, where people have asked the question, if you sequence the whole human genome in thousands or tens of thousands of people, and you look at the variants at each individual point in the genome, where are variants more likely to occur in motor neuron disease versus controls? And if you do that, you see these little peaks come up of genes for which variation is seen more often in people with ALS or motor neuron disease compared to the background. And this gives you a clue both as to where in the genome these errors are, and secondly, what these genes might be doing. And if you look at that, what you see is that there are errors, if you like, or variants in lipid metabolism, so cholesterol, interestingly, and also in the way that this vesicle-mediated transport is the way that nerve cells transport protein down these very long processes. So these are some clues that you can get from looking at populations as to why some people might be susceptible. Okay, so now I'm going to just kind of drift slightly outside of my area of expertise into evolution. I'm certainly a molecular geneticist, but I'm not a population geneticist. But it's clear that if you want to understand what makes us vulnerable to disease, genetics plays a part in whichever disease you're looking. And this famous statement here, that nothing in biology makes sense except in the light of evolution, is a useful way of thinking about this. One of the important principles of this is that in order for natural selection to operate, it's likely to operate at the level of reproductive fitness. So, for example, anything that might promote our ability to pass on our genes is likely to be selected for. It's not immediately obvious that living to the age of 120 would be selected for, unless that carries some advantage at the reproductive level. And there is all this ideas about the grandmother hypothesis, etc., whereby social groups benefit from elderly people. But it's very likely that effects occurring in early adult life which promote reproductive fitness are likely to dominate and be selected for over things that might affect the nervous system later on. So there's this concept of antagonistic pleiotropy. And what that means is that genetic variants may operate in one way in one context, but in another context, they might have quite a different effect. So here we're talking about the context of genetic variants which promote reproductive fitness in early life. And they may, in a different context, meaning in aging, carry a penalty, and there would not be any selection pressure to prevent that happening. So is it the case that diseases of the nervous system are occurring, these degenerative diseases, because the variants that lead to these actually turn out to be helpful in some way in earlier life? And that is quite a bold, unifying hypothesis. But it's an interesting way to think about this kind of problem. So we might wonder what kind of evidence might be required to actually support that kind of thing. But the point being, you know, if you develop a genetic selection process which promotes corticospinal tract evolution and all the human things that we associate with that, could that, in a proportion of people, lead to a penalty, which is a disease called amyotrophic lateral sclerosis? So what is it about humans compared to other species? Well, we have acquired in the last 4 to 5 million years, when we had a common ancestor which was largely arboreal in the forest, we have acquired bipedalism, so upright stance. We've acquired language. We've acquired manual dexterity. We have acquired something called neoteny, which means that offspring are highly vulnerable and require looking after for long periods of time. Most animals pop out, and within a short space of time, they are quasi-independent. Our children require several years of curation from their parents and indeed their grandparents. And that allows the transmission of social cognition and information, and so has probably grown in parallel with social cognition. And we have this curious post-reproductive longevity. So a chimpanzee will have its last baby in its 40s or 50s, and then it will die within 1 or 2 years. Humans are essentially the only species, with the exception of killer whales, I think, which live for decades after they stop reproducing. That's an observation. It's a very human, uniquely human characteristic. It must have some kind of explanation, but at the very least, it may fit in with this pleiotropic antagonism thing, which is that anything that is promoting reproductive and physical fitness earlier might ultimately come with a penalty, because we're living decades after we stop reproducing. So one way of perhaps looking at this is saying, well, what goes wrong in degenerative diseases of the nervous system? Well, the motor system packs up, so that's quite a human kind of characteristic. And social cognition goes wrong in dementia, and language goes wrong in dementia. Now, I mean, that's just a simplistic way of looking at it, but these very human qualities that we've acquired in recent evolution are the things that seem to go wrong in late life degenerative disease. So that's led to, you know, various hypotheses about the patterns that we see in motor neuron disease, particularly this concept of a split hand, where you get degeneration of these muscles but not these muscles, and that opposition of the finger and the thumb is a characteristically human thing, which might in itself be a sign that these recently acquired neural pathways are the most vulnerable. Okay, so can you therefore divide humanity up genetically according to genome sequence? You can do that, and you find that people of pure Southern African heritage have a much richer, greater genetic diversity. Their DNA and its variants are much more ancient, so that people in neighboring communities in sub-Saharan Africa are actually much more genetically variable than the rest of the world are, because the theory is that, I think that it's been demonstrated that the rest of the world arose from a population of people who left the Horn of Africa roughly 80,000 years ago. And so we are therefore the consequence of a relatively short period of human evolution. And if that is the case, you might wonder if ALS is arising in a population of people with particular metabolic profiles. Was that something which was acquired in that recent human evolution? And to test that, you can look at the rate of ALS in African populations, which is difficult to do, but indirectly you can do it. And there's pretty good evidence that that people of pure African origin, recent African origin that is, have half the rate of ALS of people who are descended from a population that left 80,000 years ago, suggesting that some of these things were acquired in that march out of Africa and genetic bottlenecks associated with that, which could be to do with endurance, walking, ability to tolerate starvation, thereby mobilizing cholesterol, etc., etc. Lots of different possibilities there. So we've looked here at the UK Biobank. So this is a group of 500,000 people, of whom I am one, who were called to donate information and blood about 15 years ago, and that has all been banked, and it's an incredibly valuable resource because it's a cross-section of the population with only relatively modest biases. And so we looked at this population, and we found that of those 500,000, about 500 of them have developed motion neuron disease in the last 15 years. And we have data from their blood samples from when they first attended. And what we found is that their profile of something called apolipoprotein, which is the way that the blood transports lipids like cholesterol around the body, is abnormal in a way that would promote high cholesterol, and that high cholesterol is associated with amyotrophic lateral sclerosis. Exactly how that operates at the mechanistic level is a whole other conversation, but it just fits in with this idea that people who get ALS are not metabolically random. They are perhaps in some way special. Other evidence that the sort of metabolic and athletic profile of people with ALS is different comes from this famous study of Italian footballers. This is the sort of 15-year follow-up. The original study was in 2008, I think. And it showed that the rate of ALS in Italian Serie A footballers was about 8 times what you might expect. And it was a very small sample size, and this has been followed up again and again, and it seems to be robust. Now, there's all sorts of theories. Is this because you head the ball at football? Is it because you have long bone fractures? Is it goalkeepers, midfielders, defenders, or forwards who get ALS? I mean, there's a whole load of different speculation that you can indulge in, but it would tend to suggest that people who have the capacity for exercise at a higher level than the background population may be susceptible to ALS. There is a story that military veterans may be susceptible to ALS. That's controversial. There are many confounders, but it seems to be robust. And this came out really during the Gulf War. And so the notion is that people who are physically fitter in younger adulthood have a higher risk of ALS throughout their life. And what is that all about? Well, is it that their motor neurons are more active than others? So this is a famous example of somebody with ALS called Gehrig, who was a Yankees baseball player, number 4. And if you go to anyone in the US, even though this guy died in the late 1930s, I think, they've all heard of him. And they've all heard of Lou Gehrig disease. And it has a much higher profile because we don't really have an equivalent person, with the exception of Stephen Hawking, who had a very unusual form of motor neuron disease. So the question that people ask, because of people like Lou Gehrig, is that did they sort of wear out their system by playing too hard? And this has even led some of my colleagues to recommend that patients exercise less, for which there is really absolutely no evidence whatsoever. So this is just a little insight into some of the ways that we might approach this in laboratory. So we have taken skin cells from patients in the clinic who carry genetic mutations. We can turn these into stem cells, called induced pluripotent stem cells, by reprogramming them. So then we can have these cells in a form which allows us to tell them to become motor neurons or other kinds of cells. So we can then grow these motor neurons in dish, and we can take advantage of this technique called optogenetics, where we can put into the cells a gene which originally came from green algae, which in response to light opens up a channel in the cell and makes the cell depolarize, makes the motor neuron fire off. So we've got these cells in a dish, we've given them this protein, we can then shine light on them, we can make the cells work harder by flashing a light on them. And then every time we get a flash of light, we will get a response in the cells, they will fire away. So what we're doing with this is growing these cells, trying to mature them by making them fire, but we're also connecting them up with muscle. So here are nerve cells, and they grow across these little grooves, and they connect up with muscle. And this is work being done by a research fellow in my lab called Lucy Faramond. And if the genetic mutation is present, then we can show that these cells have different forms of activity compared to people who don't carry the genetic mutation. So it could be a way of trying to connect up this apparent physical fitness, which has a much more complex explanation in terms of a network. But what does that do at the cellular level? Are these cells more vulnerable because they're firing at higher rates that make people better at exercise? This is a way in, and possibly a way that we might find drugs that might in some way help prevent this from causing damage. So I'm just going to finish with a little bit more speculation by saying, well, what do we do about all of this? Because understanding why people get disease is really the key. And we are quite a long way off from really understanding everything about amyotrophic lateral sclerosis. But our model really is that people are born with a kind of susceptibility profile. This is different in all of us. So in principle, we could all get ALS, and 1 in 350 of us will. But actually, your genetic background is really key, and that may also affect the way in which the brain develops, the way it connects up as a network. And the question is, are you losing function inside your nervous system gradually over many decades until you hit this dotted line here, which is when things start going wrong? Or does it all happen rather more quickly? We don't know the answer to that. But we think there is a phase which we call the preclinical phase. So in this phase, you're completely normal, you're susceptible but normal. Here, you're starting to go wrong, but your nervous system is compensating. And so nothing is apparent. No one can tell there's anything going on. And then when that compensation gets used up and you decompensate, you start to develop weakness, progressive weakness, until unfortunately you die. And this usually takes a few years. This is the clinical phase. All of the clinical trials we do at the moment are being done here, because that's when people present to us. And I can honestly say that's one of the major reasons why we may not be making progress, because it might be just too challenging, really, to stop the disease once it's started. And what we really want to do is to work in this space here, which I've called the ideal translational space. That means turning laboratory information into drugs that work. So that's the space we want to work in. And how might we do that? Well, we can take people who've got a genetic mutation and we can ask their relatives to help us, because they're all at 50%. Their children and siblings are at 50% of risk of carrying the same mutation. If they are willing to come along and help us with our research by giving samples, we can track them over many years and see whether we can pick up the very first signs in their blood and in their spinal fluid of the disease. So that's one of the projects that we're also pursuing.